Okay, I just wanted to kind of frame today's talk. I will start out right from the bat that I am not an expert in DTI and I have not run DTI. But the great thing about this meeting is that we can come together as a group and talk about issues and troubleshooting and how we would kind of approach this. So that is how I'm framing today's talk. So luckily, Andy is an expert here, and I kind of force them into that. So he doesn't have to, I know he's currently eating, and maybe if he has time, he could talk a little bit more about DTI. So we have some
Sure thing.
great experts. But I also thought it would be helpful, especially, for grad students or maybe, people who maybe have expertise in something else who want to tackle DTI. I think it can also be helpful because I think we are in the same boat where it's something we want to learn about. So today, I think it's kind of geared for those kind of people that, if you want to want to do this, but you don't know how, you don't know where to start. That's what I'm hoping I can do to help you, and then give you resources to do more of the specific things. So I think it'll be helpful for the people that have done DTI to talk more in the discussion about things that they have found helpful. So um, and specifically, so my KO1 study, the neuromod study, that's the real-time study that Adrian mentioned. So I am collecting DTI data for that study because connectivity as well as developmental and sex differences is a key piece of that. So I was in a meeting, and I believe it was Chris Monk said, well, if you're looking at connectivity, you might as well go ahead and collect the DTI data. And I thought that's a good idea, especially, starting out. So we're collecting it, but we're not really sure what to do with it yet. And then I've been working with an fMRI analyst in our group, Ryan Lash, who I think he is on a snowboarding trip right now. I saw him on video. So he's very dedicated. He is on this call on a mountain right now, snowboarding. So if he's available, he might chime in too with some of these comments. But so also, as Adrian mentioned, I'm working with the Michigan longitudinal study. And I've talked with Mary Heitzig as well as Mary Souls and again Ryan about, okay, we've collected this DTI data. Can we do something with it? And people are not really sure where to start, and it's on the back burner, so to speak, because we know how to do the other stuff, and so we're a lot more engaged in that. And then also, I'm working on the ABCD study, and DTI is also collected here. So being part of these studies, I know that there is DTI, and so it's something I would like to pursue and kind of think about how we can start analyzing that data. Just to start, I'd like to go over the basics of DTI. What is it? You probably have seen these fancy images before. They're very colorful. I think when I was starting out in fMRI research, I put one of these on my screensaver, well, this is cool. I want to do this, but there's a lot more that goes into it than just getting these pretty images. So this is a structural imaging technique, and it measures both the orientation as well as the direction of white matter fiber tracks. So those are basically the myelinated axons. And it does this by mapping the diffusion of water molecules. The nice thing about this is it uses existing MRI technology. So if you're already running an fMRI study at the lab, you basically just add on oh, can we also get, a DTI? So it's pretty easy to add on the front end if you already have an existing study. And then usually what we have them do is watch a movie or a video clip. That's what ABC does. You don't have to think about anything or even, resting state where you have them usually look at a fixated cross. For DTI, it doesn't really matter for that. And it's important to keep in mind, this is from a talk that Robert, I kind of quoted him here about Robert Welsh, who is a DTI expert here at Michigan. He's given several talks, and I thought it was important to point out that this is a modeled interpretation of fiber connectivity. So we're not really measuring the axons, but what we are measuring is the water diffusion. And it's important to note that we believe that that is being impeded by these tissue boundaries and membranes. So it's really this kind of indirect measure of fiber connectivity, but it's the best we can do with live in vivo subjects. And I just wanted to visualize here the structural versus functional connectivity and kind of where DTI falls within that. There's also DSI, which is a little bit more advanced method of kind of tying into DTI, but I won't get into that today. And just to recap, this is kind of psychology 101, but I think for people that don't do brain imaging research, it's kind of good just to go over this again, what gray and white matter is. So gray matter is about 40% of the brain matter. These are neuronal cell bodies. And this is really where the processing occurs. So, with functional data, we want to focus in on the gray matter typically, and then we regress out white matter and cerebral spinal fluid, for example, sometimes. And white matter actually comprises more of the brain than gray matter, so about 60%. And this is where the communication with the other gray matter areas occurs. And the myelin aids in the communication. So you may have learned about, what myelin does, and it basically facilitates more effective communication with the brain and also a greater speed. It's very highly ordered. And important to DTI, there's a higher water diffusivity parallel, so it runs with the nerve fibers in the white matter of the brain. And these are some neat images of what it looks like at a very minute scale. So what we're talking about with the microarchitecture of the brain. So these are some cool images, again, from Robert Walsh's talk he gave. Another, kind of going back to high school science, what is diffusion? And this is important just to make sure we're aware of what this is, because this is, tying into what we're measuring in the brain here with DTI. So it describes random motion of molecules, and this is on a microscopic scale from higher, which is on the left, we see here on this image, to the lower concentration, and it's a passive process. And as I mentioned before, we believe that there are these barriers that impede the movement of the water in the brain, which is how we then measure these white matter tracts. So you can see with the blue dots, that's an example of what cerebral spinal fluid looks like. So it's more of that random motion, but the white matter, you can see it's more constrained. And these are some key features of DTI that will carry over through the rest of my talk, especially for terminology. So the isotropic, this is on the left side of the two blocks over there, that column on the left of the image here. It's describes an absence of barriers, and diffusion is equal in all directions. And as we saw in the prior image, an example of that is like CSF or water in a glass of pure water. Versus anisotropic, which is the presence of barriers. It's directionally dependent, and the movement is along the fiber tracts. And also another really important part of DTI is understanding this diffusion tensor model. And I'm not going to get into it in too much detail here, but it's important to note that it's a kind of the key pieces here, that it's a three-dimensional Gaussian model. It's a 3x3 symmetrical matrix with the three orthogonal eigenvectors. And these are a local fiber coordinate system. And you can see here, that's the bottom circles or the bottom squares down here in the figure is where that is shown. And so these three eigenvalues give the diffusivity and the direction of each eigenvalue. So it's important to know these kind of the basic understanding of the diffusion tensor before you get into the analyses, because this will definitely come into play as we talk a little bit more. And so for the origins of DTI, curious if you notice anything strange about this picture here. What is in the scanner here? So that is a squid. And so pre-DTI, the orientation of these axons in tissue had to be known. And so they could only use fixed samples. For example, the axon of a giant squid could be scanned, which is really fascinating. I didn't know about this until I was looking more into DTI. And areas of research that's been applied include, but not limited to, autism, schizophrenia, Alzheimer's, traumatic brain injury, that's a big one that's been looked at, and substance use, which is in my area of interest, as well also in developmental change and aging. And it's the animal theme here, sticking with that. It's even been used in dolphins. So this was post-mortem, but they looked at the sensory and motor system in dolphins. So there's a fairly wide application of DTI in both human and non-human samples. Before you get too excited about the possibilities of DTI, I think it's important to note starting out that only one fiber direction per voxel can be modeled at a time. It's also very sensitive to motion. And this is important when we're thinking about young samples. Like this has come up a lot with ABC sample. They're starting at 9 and 10-year-olds. Inherently, just younger samples, they move around a little bit more. And it's susceptible to these artifacts that are called eddy currents, and they're caused by this rapid switching of the diffusion gradients. And that's what it typically looks like on the image. And then interpreting the connectivity, the tractography, is something to consider too, because there are areas of the brain that have 2 or even more differently oriented fiber bundles within the same voxel. And this is referred to as crossing, diverging, or kissing fibers. So these are some things you need to think about, especially when you're interpreting your findings. All right, so moving on to the DTI data acquisition. I mentioned in the description of what I was going to talk today about was the workflow. And this is in one of the articles that I sent out to look at before today's talk. I think this is a really good visualization of where to start, and then how to take it like one piece at a time, because there are a lot of pieces. And so I think breaking it down like this is very helpful. And so here's an example of the ABCD study workflow here. And I tried to block out kind of the important parts here. So if you're familiar with ABCD data, there's a lot of information being collected. And so here's the pre-processing steps over here on the left, which you can see, and then the processing pipeline input and outputs over here on the right. So you can see the different kind of files that go in and the structure of those, and then all the different arrows and pathways, and to get down to where you want to get to the DTI analysis. So before you get to DTI analysis, there's quite a bit involved. I think Rich Gonzalez said, like, bad data in, bad data out. Like, it's a really good way to think about it. You might be thinking you're running your analyses correctly, but if the data isn't corrected for these important artifacts and issues early on, you're not going to get accurate results. So it's really important to start at the very beginning and make sure you're following those appropriate steps. So data quality is really important. It's also important to have a high signal to noise ratio in order to make sure that your parameters and image accuracy are optimized. And you will get greater precision if you have a higher field strength, and stronger gradients improve the signal for DTI. Also, parallel imaging using the RF coils with multiple receiver channels is another way to lower the acquisition times and image distortions. Another thing to consider is the scan will cause vibrations in the scanner bed. So we tell our participants to expect that. We don't want them to think like, oh no, hit the button, emergency, like it's breaking. That's normal. It's gonna shake a little bit more and have more vibrations. But those vibrations, though, can contribute to artifacts. So it's something to consider, when you're doing the quality control. And the average scan time is about 5 to 8 minutes. And that's why it's nice to usually show a video clip. You don't want to do anything that's too funny. You don't want them like moving around and laughing in there, but just, kind of a neutral video. Or I think we show like Harry Potter in ours, maybe, but just so we just want to make sure they're comfortable in there to sit in there for the time. So we don't want, this is the optimization part where you want to get as much data as you can at the shortest amount of time possible to get that data, as with any imaging task, really. Okay, so going into the quality control and pre-processing. Some issues that come into play with quality control are these, as I mentioned, that it's mechanically demanding, this fast gradient switching and the echoplanar imaging, and getting into more of these common artifacts you have to watch out for. It seems like with DTI, the recurring theme here is the eddy currents and the motion artifacts. Some other things, though, to consider are the slice and gradient wise inconsistencies. I think it's good here to see, to visualize these. Um, so looking at different examples of what to look out for these artifacts is helpful. There's also some very helpful resources out there on the web. So DTIPrep is one that I recommend. I can share these slides too. I don't know if you have a space, Adrian, to share slides. Okay, great. So yeah, I can put this on here with these links. So this is one, and it has quite a bit of information through here. I just took a screenshot of the main study page. But there are, things, resources you can download here, and there's, I believe there's like a forum you can talk about issues as well that you might come across. This is the DTI prep workflow, and as with all things DTI, it's complicated, but again, I think just taking it one step at a time is very important. So I won't go into the details of what each of these involves, but it does tie into some things we've talked about before. So just making sure that you don't go on to the next step until you've thoroughly completed the prior step. Another option is Tortoise. So this is a software package that is useful for processing diffusion MRI data. There are 4 main modules in here. There's DiffPrep. This is used for example, motion and eddy current distortions, like I had talked about. DR Buddy, I don't know if I'm saying these right. This is for the EPI distortion correction in particular. DiffCalc for tensor fitting, error analysis, etc., and DR Tamis. So this is for looking, for example, at inter-subject registration and template creation. Okay, and so once you have all that data, look through the quality control and the pre-processing, another key part of this obviously is the data processing and visualization. And this is tying back into those fancy images, and how do we think about how we're displaying the DTI, the white matter tracts in the brain. And there are a few options here. So tensor estimation occurs at the voxel level. And so it's the minimum number of these diffusion weighted images required to do this is 6. And the typically used methods to do the tensor estimation is OLS. This is the most common, as well as the weighted linearly squares and non-linearly squares. And this is, I'm referring again to the article that I sent to you to look at. So you can look a little bit more in the detail here, but I'm just giving an overview. In terms of displaying the data, one option is the scalar. So this is the tensor information from a single number that corresponds with the diffusion magnitude or the anisotropy. When you look at DTI papers, they talk about these terms quite a bit. And so I think when you're learning how to do DTI, I would just read through a lot of relatively recent DTI papers and good journals that are in your area of interest, just to see, what they're reporting with their methods, I think is really helpful. And then you can see how they're interpreting their results. And so these are the main ways that people report these results. So the mean diffusion, MD, is the average of the tensor's eigenvalues. So this is the total amount of diffusion in a voxel. The FA, the fractional anisotropy, I would say this one would maybe be like the most common result that you typically see, or one of the most at least. So this is the fraction of anisotropy diffusion. This is the difference between the ellipsoid shape versus a perfect sphere. And I'm going to show you what that looks like here. So this ranges from 0 to 1, 0 being isotropic and 1 fully anisotropic. So if you remember back the square images that I had earlier on, you can see that the shape of the ellipsoid. And so, like, for example, this is maybe what, like a FA of 0.9 would look like. It would be, you know, a lot more kind of flattened down. There's also a apparent diffusion coefficient, the ADC, the magnitude of diffusion within a tissue, and the axial diffusivity. This is focused on the main axis of diffusion. And then the radial diffusivity. So this is in the transverse direction. Some important things to consider here is that these diffusion software, acquisition parameters, processing, and analysis methods. And so it's really important that you report everything you're doing and you try to be as transparent as possible. So, so in terms of, you know, replicating, you can kind of follow the same pattern and you will get more consistent results. Because this does have important implications if you're looking at different comparisons with other studies or multi-site studies, which I show here as an example. So in ABCD, there's 21 sites across the US. And so if we have all these different sites, how are we harmonizing the DTI measurements across scanners? That's really important. So there's actually a whole paper that has been written. And if you're interested in looking at DTI data and ABCD, I would definitely check this out. And again, you know, just the transparency is really key here, just making sure that you're going through each step that you took to complete your DTI analyses. All right, now we're getting into the pretty colorful images part of DTI. This option here is, so putting the tensor information to 4 numbers, and this gives you the color and brightness value, with blue being superior to inferior, red left to right, green anterior to posterior, and then the value of the brightness is a indicator for the tensor anisotropy. These are called glyphs, and they're a 3D representation of the major eigenvector whole tensor. And the images, the first one I showed you, so this is tractography, and this is an estimated course of the white matter tracts. And this is, it relies on the principal diffusion direction, and the colors here are automatically assigned based on an atlas-based tractography segmentation. Okay, so going into the quantitative analysis here. So if you want to look at, you know, which is what pretty much all of us want to do, is eventually to look at individual and group level differences here, is it's, you know, extracting the summary measures based on anatomical regions or whole brain, which is pretty much what we do for all types of MRI data. One approach is doing an ROI analysis. And so one option is to do a manual, a priori specific region selection, or you could do an automated parcellations. And so this is usually a way to measure like the MD or FA from those specific ROIs. The histogram analysis. Unlike the ROI analysis, this is automated, and a histogram of the parameters such as MD and FA are really useful to look at the groups and compare them by statistical analyses. Okay, here's an example ROI paper, ROI analysis paper. They looked at white matter, integrity, substance use, and risk-taking in adolescents. And I'm gonna, before I go on, I'm gonna mention Robert Welsh again. He does not like the term white matter integrity, and I think there's a camp of people in DTI that don't like that because it's not exactly what we're measuring. So something that might be a different way of saying that might be the white matter coherence, white matter microarchitecture. So that's something to keep in mind too, just being aware of the terminology there. So in this study, they looked at bilateral white matter regions that were associated with higher order cognitive functioning, as well as these fronto-subcortical and cortical limbal projections. And for this study, they did have 4 ROIs that they selected in advance. And they used a binary parcellation map to look at the FI coefficients for each ROI. So the main findings here, and you can see this is one way that's fairly common to report the FA is through these, these images where the plots of how you're associating the FA with some kind of outcome or predictor variable. So here they found that lower FA at baseline and specifically in the fornix, which is the top figure, and the corona radiata predicted follow-up substance use 1.5 years later. And then, so they also found that this predictive association was found above and beyond variability accounted for several factors, including baseline risk-taking, emotional functioning, and family history of alcohol use disorder. Okay, so now this is the voxel-based analysis, the VBA. Voxel-based is used to compare local anisotropy values at the whole brain level between participants. And you can see a comparison here on the tract-based on the left versus the voxel-based on the right. When you're doing this, it's really important to correct for multiple comparisons. I think there's been several methods that talks about how the importance of correcting for multiple comparisons. And as with other types of imaging analysis, it's important to do that for this as well. One way to do this is by using an atlas segmentation method to focus on a particular area of interest. It's also important to normalize the data here since DTI is so directional and topographical. And then multimodal studies. This is something in the Heitzig lab we've been talking a lot about, especially with the Michigan longitudinal study. We've got, this great array of data, fMRI, like functional task data and resting state and DTI. Is that something we can look at all these different pieces of the neural functional puzzle, so to speak, to better understand maybe, individual group differences on some kind of indicator. And so it is possible to do this. And one way to do this is using the MRI or the T1 weighted mask segmentations or parcellations in DTI to extract these combined structural and diffusion data. Again, this could be a whole separate talk, so I won't get into this, but it is discussed in the example article I sent. And there are some good reference papers, and I can help direct you to those if you want. And perhaps Andy knows a little bit more about this kind of the multimodal approach of DTI as well. And it's important if you're doing this that the ROIs are in the same reference space as the DTI data. That ties into interpreting. So you want to make sure that you're interpreting all these separate pieces in a similar way when you report your findings. Okay, so I'm going to go over this example study. It wasn't one I included in the reference articles. Those were more like review papers. But I wanted to walk through this. This is an example of what you might want to look for when you are starting to explore these example DTI studies. So this was done by Bealuna's group to look at developmental stages and sex differences of white matter and behavioral development through adolescence through a longitudinal DTI study. So basically, they just characterized the timing of white matter development and looked at how these other correlates were involved in the developmental trajectories of white matter. To give a little bit of background for the developmental people that do any kind of neurodevelopmental focus in their research, you may have seen these images. This is Jake Eads' kind of seminal work that he did. Finding that there seems to be a linear increase in white matter across development, so from like adolescence into early adulthood, and increasing less in females than males. So there's a sex difference, it seems like, as well. And for gray matter, we see this pre-adolescent increase and then post-adolescent decrease. And the sex differences we see here are typically associated with the earlier pubertal timing in females. Okay, so this example study, they had 128 8 to 29-year-olds, and they were scanned either from anywhere from 1 to 5 times. For their data acquisition, the data diffusion gradient was applied in 6 directions, averaged over 14 repetitions. And they did this to increase signal to noise ratio because, as we talked about, that's really important with DTI. So it's important to implement strategies to be able to do that. For their pre-processing, they use DTIPrep, which is what I showed you earlier as a good way to run your pre-processing. And this automatically identified and corrected motion and scanner-induced artifacts. And then they also looked at the images slice by slice, so taking that one step further. For their processing, they used FSL. They converted the raw DICOM images into NIFTY, created mask using brain extraction, and they also corrected for head motion and eddy current distortion, because those are the 2 biggies with DTI data of things you need to account for at the quality control and processing level. And then they use the tensor model to fit their images. And then beyond that, they also ran some group analyses looking at FA, AD, and RD, and they adjusted these for registration-related artifacts and normalized them to a template. And again, normalizing them so that it can be kind of examined across the different measures more accurately. And they used an FA skeleton generated and thresholded the FA of more than 0.2. And remember, we looked at those different shapes, and so that is fairly lenient, I would say, FA threshold. So for their ROI analyses, they looked at core white matter tracts and regions adjacent to gray matter, and then the non-DTI things that they looked at as well. So the developmental effects, interactions with sex and behavior. This was done through an anti-saccade task as a correlate of cognitive control. And they also looked at hemispheric differences. Their main findings through this study for FA was that they found an increase with age. This image here, you can see the FA is on the left column, RD is in the middle, and then AD, and then the percent change per year legend is over on the far right. So after adolescence, and then the age is at the bottom too, so it goes from 10 to 25. So they found that after adolescence, all the way through into adulthood, there was continued growth, especially in the frontal and limbic tracts. For RD, there were decreases with age, and then axial diffusivity, the AD, there were decreases with age as well. And the timing and developmental change was typically orthogonal to the FA. So you can see here an example of how you can look at several of these different measures of DTI across people in like the same figure, for example, because they are all related. In relation to sex differences, they found greater change in FA and RD in males, but no significant differences in AD. For the behavioral development, the earlier maturation of white matter was related to a more efficient network integration, and delayed maturation was related to a weaker cognitive control. And so they believe that this, especially the protracted maturation of the cingulum, was contributing to improvements in cognitive control. As a reminder new information for those who have never heard of the cingulum, it's basically white matter fibers that are going from the cingulate gyrus to the entorhinal cortex. And it aids in it's involved in communication between the limbic system and also the ACC to integrate other areas of the brain, specifically involved in inhibition. So they found kind of in sum, taking all this information together, that the white matter maturation seems to occur in a hierarchical manner, and these pathways suggests that this executive control matures through adolescence, and specifically pathways that are related to socio-emotional processing also continue even beyond adolescence into the 20s. And with these sex differences, they found that males showed the later maturation than females. And they tied this into other different kind of sex differences we see with sex-related vulnerabilities to psychiatric disorders such as depression. And these outcomes were influenced by the timing of these different neurodevelopmental processes. So this is a good example of a developmental focused DTI paper that is worthwhile checking out. Okay, so now getting into some questions for the group. I think we have about 15 minutes left. So and if there's any specific questions about DTI methods that I can, if you want me to go back to any slides or work through anything, or if not, we can go on to some specific questions that I have as well. So I guess, are there any general questions before we get into these specific questions? No? Okay. I don't know if you have a chat function too. If someone thinks of something, they can put it there. Oop, let me try.
Yep, I'll keep monitoring, Megan.
Okay, no, that's fine. And I don't know if for those who have, or how many have conducted DTI before, but for those of you that have, what are some issues that you've come across, either things that I brought up or maybe some additional issues, and then how have you dealt with them?
I guess I have a question for you, Megan, as well, maybe a little bit before this, which is, you've provided just such a great overview of all the different steps, consideration, points of this analysis pipeline, and maybe for those of us who aren't using DTI in the group, I guess what, from your perspective, what are the advantages of it? Like, despite the complexity, the potential limitations of things that can be brought up, I guess, why have you and your group been so interested in diving into it? What can it tell us that we can't learn in other ways?
I think we've been focused so much on function, and I in neuroimaging, I feel like we separate the pieces so much, but it's really a complex integrated system, and so I think the structure and the functional are really intertwined in a lot of ways, but we do separate them out for good reason. But I think it's important, if we are looking at the functional, to understand what's going on more at the structural level at the brain, to see what is perhaps coinciding with those functional differences. And in one of the articles I read, it might have been in one I included here, I think they said that the reliability of structural data is a lot better than functional data. And that's also another thing, because as we know, that functional data, unfortunately, is not very reliable. And there's been a whole area of research on this. And so I think if we are looking at developmental change, structural development, I think, has higher reliability if we're looking at the structural data versus functional as well.
Yeah, that's really helpful.
Okay. I think there was one person that was going to ask that I saw come up. Some Lee.
Yeah. Hi. Yeah, I was just, I've done some diffusion research, and I just was gonna say questions or like issues that I've come across. I feel like have been a lot of like the limits of computation and like how long it takes. I feel like has been a big barrier, because doing some of the methods, it takes sometimes, if you don't have access to great computing resources, it can take like 24 hours to analyze the data for a single subject, like continuously running. So I think that's a big issue that's come up for me, is just computational power and having access and methods that alleviate that a little bit has been really helpful. Like using the Great Lakes, or used to be the Flux cluster, was the best way for me to deal with it, because I don't think my little computer could have handled it.
Yeah. So that using more of like a server or like is helpful. How did you get access to that? Like the Flux cluster, for example?
So being a grad student affiliated with LSNA, we got it for free, just for being affiliated with LSNA. But I think you just like email them and get an account on it, and it's relatively inexpensive computing resource, I feel like. Like I said, I haven't had to pay for it, but I feel like other people on here probably have had to, and can speak to how much it actually costs. But I feel like it's pretty cost efficient, and pretty easily accessible once you have an account on there.
Yeah, there are a lot of great resources at Michigan, and I don't think people are really aware of the extent of it. So it's worth looking into, I think, before you think it's impossible, or maybe if you run into these roadblocks like the processing time, before you give up entirely. I think it is helpful to look at these different options and utilizing a kind of a heavier workload system that can handle that.
Yeah, for the cluster processing, they can get you set up with a starter account, so you can have a certain number of credits free, basically, right? So you can test out your processing, make sure it works before they actually start charging you. And then even when they start charging you, the rate isn't that high. And some departments will cover it. Like I think in Michigan Medicine, if you're part of the medicine system, then they'll give you, again, a certain number of cluster processing hours per year free. So
it's good to know. And that's available to like graduate students, postdocs, faculty, everybody?
I guess I don't know the breakdown on that. You may have to check with them. I think that they'll give that at least the starter testing out, credits to anyone. But in terms of like, whether the Michigan Medicine one applies to everyone or that's just faculty, I don't know. So.
Yeah, definitely worth looking into. So who would you contact about that? Would you start with maybe someone in the fMRI lab to direct you to a contact person, or?
I think I just contacted ARC directly. If I find a contact, I'll pop it in the chat.
Okay.
Yeah, I think if you just Google like Great Lakes, University of Michigan, that'll probably have like a step for how to get an account.
Okay.
They have like a whole tutorial.
Wonderful.
Yeah, that brings up a question for like the larger data sets. Do you know, Megan, with like ABCD, are they doing any like standard pre-processing of their DTI data before it's released, or in what form are those data released? We talked about ABCD a couple weeks ago here as part of this group. So, I know a little bit about what Lee went through in terms of, computing for her sample, which wasn't the thousands of folks that, that are available in ABCD. Do you know? So do you know anything about like how the DTI data and ABCD are being released, or do we all need to dig a little bit more?
I don't recall from that article I read about the harmonization of DTI data, but I would assume it would probably be in that article. If they're already talking about how they're harmonizing data, I would think it would be there. But that's been an issue with like the functional data, for example. We've been working with Chandra Sirpada and Mike Ingstad we've been just getting the raw functional and resting state data, because we don't, we just kind of want to do it on our end, and do it in a kind of a different way that maybe we are more used to, or like we maybe feel like we're taking a few extra steps So I think if you do rely on what they've done, I think it's important to, again, with that transparency, to know what they've done and gone into it. And then you can decide if it's more worthwhile just to get the raw files and do it on your end, or if to take, their files and what they're doing. I did I show that image of the pipeline that they're doing. So I could look back at that information to see like if all that's done for the data that they give you for the DTI data, or if that's something you need to do on your own. So but that's something really important to consider. If you're just going to be, they're going to dump this massive amount of data you don't know what to do with, or maybe you can at least have some quality control or pre-processing would be helpful.
Yeah, I'm thinking of what you said about like checking slice by slice for artifacts, and I know there's some level of like manual checking that goes on with this work, but thinking of that at the scale of ABCD is kind of mind blowing.
Yeah, and I don't know if ABCD is the first large scale study that has done DTI, if there's any precedent to that. How are we accounting for these really important quality control issues on a massive scale like this? What kind of big data approaches are we using? I assume they've thought of that with ABCD, but it's worth like digging into and looking into what they've done. And I don't know, Andy, if you're willing to share just some where people have come to you with when they've done DTI with like, what are their typical problems that they deal with? Maybe someone just starting out on DTI.
The typical problem I usually see is dealing with distortions, because diffusion in particular is very susceptible to, say, motion artifacts, as well as these things called eddy currents. And the best way to remedy that, I found, and what you'll need to acquire when you're at the scanner, is something called a reverse phase encoded image, which I believe is a default here at Michigan these days. But other people I've worked with other places, they only collect one diffusion data set that has phase encoding in one direction. And all you need is just one volume in the opposite direction to unwarp a lot of the distortions, and your data will be much better, much more valid. That's one thing. Other typical problems. So what I do with regards to, say, checking individual slices, I usually don't do that manually. The approach that I do, I use a software package called Tracts. And what it will do is it will flag individual slices that seem to be corrupted by, say, motion or something else. And you can write a script. I have a script available on my website, which just simply does a calculation, what percentage of slices are corrupted. And according to the developers, if it's 10% or more, then that's grounds for throwing it out. That seems a little too clean of a number for me, but there you have it. So for me, that's a way to have like a single number that gives me an idea about whether that subject is a problem or not, without having to do checking every slice, which might be prohibitive. Might not have enough time to do that.
Mm-hmm.
Those are the two main things that stick out to me.
Great, thank you.
Yeah.
Tracts. And is that, so that's just something you can download for free, or?
Yeah, it's for free. Last summer, I wrote a walkthrough, which is now in my ebook. It's closely patterned on, her name is Marlene Tejedal. She's University of Regensburg, I believe. And she wrote a tutorial about Tracts. I've, more or less followed the same steps. And that in turn is based on the Tracts documentation, but kind of adapted it more towards data sets that I typically work with. And there are a few things that I do, a few options I use that she doesn't. It's simply my own preference. But yeah, it is available. I can send the link if people want.
Wonderful. Yeah, and it ties into what I was going to bring up next to all these different DTI tools, and some of them can be a little overwhelming, and then you're like, well, what do I use? And so here's a table, for example, of different options and what can be used for.
Right.
Yeah, and then we kind of already talked about the white matter integrity issue, but yeah, I want to promote Andy. We've got a great resource here at Michigan, and yeah, his ebook. And your ebook is out now, or is it?
It's been out for a while. It's kind of
Okay.
it's constantly under development because I'm adding new
Work in progress.
modules. But yeah, MR Tricks is on there, and I have a short introduction about diffusion. One more thing I was going to bring up is that so with something like MR Tricks, there are different ways to analyze data. And so you talked about FA, which is very common. It's very popular and people understand it. MR Tricks does something slightly different. You calculate streamlines that connect different parcellations. And basically it's calculating the relative density of streamlines, say between groups or before and after intervention, for example, as a way to measure the overall connectivity between different regions. They have other things as well, but that's their main analysis that you can do. So as far as interpretation, I mean, it's kind of up to you. Like, did it grow these connections between different regions? You can put them in a connectome, a huge correlation matrix, and then overlay that on something like functional connectivity to see how well they correlate, whether structural and functional are aligned in some way that might give you insight into some kind of disorder, disruption, and so on. So I found it really useful.
Great. Have there been many published studies that have used MR Tricks, or is it
surprisingly not that many that I've been able to find. The group that makes MR Tricks will publish technical papers when they release a new method, for example. But I haven't really collected a library of people outside that group who have used it for research questions. So that's a good thing to look into. Maybe I'm just not as aware about it, but it should be pretty simple to find, right?
Yeah.
So
Yeah, I was just curious that
MR Tricks, yeah.
kind of see like what methods they use to apply the MR Tricks. Okay. Great. Thank you. That was all helpful. So, well, thank you all. Hopefully you got, some things out of it, an overview. At least I could hopefully direct you in different directions if you want to try to do DTI yourselves.
Thanks so much, Megan. Really appreciate all the resources, and it's so helpful to have a talk where we feel like we're learning. You're like a couple steps ahead of us and telling us what you needed to know and learn along the way, and kind of bringing folks along. And that's really helpful for a lot of us. So really glad you did this today. Thanks so much for your time.
Thank you.